Well, nice to see everybody here today. This is Phil Calderone hanging out for Sunny Shell again on the sunny. I keep wanting to try to say the sunny side, the sunny spot on Skeptic Haven channel. Uh, we have a medical show for you today with somebody that I think you know quite well in the atheist community, particularly from the ACA. Um, our topic today is cancer nanotechnology treatment with MD Aware. You've seen him talk a lot about atheist issues, and we're going to talk about that a little bit today, but dominantly concentrate on the topic of nanotechnology in medical therapy. So, uh, please like and subscribe right now while we're thinking of it, right up front, please. We always appreciate and like to have more subscribers. It gives us more ability. So, right this second, if you can, hit that subscribe button, like the video, and don't forget if you're interested to consider supporting us with a Patreon pledge. All of our channels are slash Skeptic Haven on all the social networks. So let's get this thing started, and a little bit of intro for you. Well, welcome, MD Aware. Thank you for showing up today.
Oh yeah, it's my pleasure to be here. Thanks for the invitation. Should be fun.
I'm looking forward to this one. There's always so much to learn on every topic, and this one, I know like this much about it, and I don't know how much the audience knows about it. So, um, I think we're going to be learning something here today. Let's start by telling the audience about yourself. I've seen you on ACA and things, but that it's not related to medicine per se. So give us a little intro of yourself here in your history.
Sure. So I live in Canada, born, raised, educated, everything. I sort of wanted to be a physician for a long time, like a lot of people, I think, who ended up in medicine did. And that led me into an undergraduate degree in biology. So I did like your sort of standard life sciences undergrad. I got very, very interested in science as a process as a result of that. It's kind of interesting because I think a lot of people who go into like life sciences, the study of life sciences, are using it as a means to the end of becoming a physician. And that's certainly how it started for me. And I think that's probably how it was for 80 to 90% of my class. But for me, the scientific process really fascinated me and grabbed me. And partly as a result of the fact that I went to a relatively small university for undergrad, I was able to do a very broad scientific background. So I had predominantly biology courses, but I had a fairly even split in terms of like chemistry, organic chemistry, biochemistry, physics,
Nice.
astronomy, psychology. So I sort of covered all of those bases, and it gave me a really nice broad basis in science, which I thought was really nice. My undergraduate thesis was related to breast cancer. And then after that, I went to medical school, and I did some research there in breast cancer as well. After that, I I ended up in Toronto for general surgery for residency. So that's what I'm doing now. And so I did 2 clinical years of training in general surgery, which, if you don't know what general surgery is, we basically deal with all of the soft tissues, sort of like from your neck to your pelvis. And basically anything that the other surgical specialties don't do. So we will do anything from like thyroid cancers and thyroid operations. We do breast operations, esophagus, stomach, all of the gastrointestinal tract.
Wow.
We do liver, gallbladder, pancreas. Trauma surgery is our domain as well. People can go off from general surgery to do like thoracic, so they deal with lung stuff, or vascular surgery potentially, and dealing with blood vessels. And we do a lot of cancer research, or a lot of cancer surgery as well.
I
So
have,
that
I
for
have,
2
I have a
years.
little
Yeah,
bit
go
of
ahead.
a goofy question here.
No problem.
Did your cadaver have a name? Did you give your cadaver a name?
So when in med school we did do cadaver dissections. They obviously had names. I don't think that we were told them, because they're supposed to be anonymous, and we
Well, I
try to be
mean,
very respectful of them. But
like I don't know, whatever. So,
yeah, it's a tricky thing, because, like I said, we try to be respectful
because of the respect issue. Okay. Oh,
of
nice.
the donation and the sacrifice, and
Yeah.
because it's at some point you have to acknowledge it's someone's
Yeah.
grandfather or parent or whatever, right?
Yeah.
And so you try to learn what you can, but the things that I think are very distinguishing are the sort of like interesting findings you might see. So we had, like, I don't know, maybe 4 people to a cadaver in medical school, and you would know, like, oh, this person died of lung cancer, and you can go and look and find all the tumors that might be in the body, or this person had like a previous coronary angioplasty, and so you look and you dissect out their coronary arteries, and you find the stents that were placed there. So you get to know the patient or the cadavers not by name, but by, like, what their pathologies are and what interesting findings you see in them. So that's kind of how we identified them.
It's interesting how much. I mean, when I was in pharmacy school, we went to a dissection. Well, it was after the dissection, with a pathologist, and she showed us a lung from a heavy smoker, and boy, that does not leave your head. The difference between a tar-filled lung versus a clear one.
I mean,
It's something that
even the effect of
away.
living in a city is pretty remarkable. You can see the difference when you look at the lung. We wouldn't say that it's clinically significant. It doesn't cause any, problems necessarily, but you can certainly see visually the deposits of, more particulates in the lung parenchyma in the lungs of someone who's living in the city their whole life versus someone who lives in the country, and they have, probably better air quality.
I saw you have an interest in evolutionary biology. That was going to be a backup for you, or?
Yeah, that's right. So because I got so interested in undergrad in, like, scientific method, and I was studying biology, and admittedly part of it was, my journey into atheism was happening at the same time, and sort of like as a result of that education. So I started reading Dawkins. I read, like, The God Delusion, and that was really good, but I saw he was an evolutionary biologist. And so I read The Selfish Gene, which is, like, very, very, a seminal work in evolutionary biology. A great book. And that got me really interested in evolutionary biology. And so, I was going to do, I was accepted to a master's program and everything, and ready to go when I got my acceptance to medical school. And then I had to make a decision, and I ended up having to make up a lame excuse as to why I couldn't do the master's degree, because I didn't want to be like, oh, hey, professor who accepted me and, like, took me on a tour of his lab and bought me lunch and, like, all this stuff. Sorry, I'm gonna go with the med school. Like, I just couldn't do it to him. So I felt bad about it, but it's one of those things where you wonder what your life might have been like if you'd done the other thing, made the other choice. So maybe there's a parallel version of me somewhere who is an evolutionary biologist instead of a physician.
Well, there's a bit of a parallel of you right here, right? Because, I mean, this has got to come up fairly often on your online discussions, Talk Ethan and such. I would think on occasion. I mean, you've got a lot of shows. You've got a lot of episodes that you're in, I've seen.
Well, yeah, I've done the tour around the ACA. My home show is Truth Wanted, which I like. I mean, I think Truth Wanted is the most broad. So we will take calls not just about like atheism and theism and like religion related stuff, but we've like, I think my very first show that I did for the ACA, we got a call from someone who thought he was a time traveler, and we got a call from someone who wanted to talk about like cryogenics. And so, you know, that show runs the gamut of subjects, and it's useful to have like a broad scientific background to make sense of that stuff.
Yeah.
You know, apart from all of the, you know, you still get a lot of religious based callers to those shows.
So, sorry, I cut you off in your career a little bit there. So, how'd you push into surgery then? How'd that interest come to be?
So I did 2 years of clinical training in surgery, but I knew I wanted to have science as a part of my career. And so we have a program called the Surgeon Scientist Training Program, where if you are in a residency, in a surgical residency, you can take time off of your clinical training and do a graduate degree. So I did that. And so the past 3 years, I've been working on a PhD in biomedical engineering. I just defended my thesis recently. So I'll be going back to clinical training soon. But basically, for the past 3 years, I've been working on developing cancer nanomedicines for potential applications, and really trying to bring them out of the laboratory and into the clinic where we can use them. Because obviously, I am uniquely placed compared to most scientists in that I can study a thing in a lab and then also bring it into clinic and treat patients with it. Yeah. So that's where I am now and sort of where I'm going.
It's a kind of a fascinating thing to me, because you know, when you go into medicine of any type, I mean, there's a certain amount of repetition in what you do, right?
Definitely.
But when you've got a research element, it's a mind broadening thing, and that I would think that really helps motivation and long-term interest and excitement of discovery. And
and I mean, I think it makes, I mean, biased opinion, but I would say that makes you a better physician because it makes you stay on top of the research. I think that physicians in general, but surgeons in particular, are very apt to get set in their ways, because it's sort of such a, there's a lot of art to surgery still, and so people learn one way to do it, and they're sort of scared to try something new in case they do something wrong and hurt a patient. So I think that, you know, being able to stay on top of the research, keep up with new developments, and I mean, I think doing research keeps you open-minded, and I think that's really important to be a good physician, but also key to being a good scientist.
How do you stay on top of research? I mean, you know, my study as a layperson really, but is belief and psychology and neurology around that, but the volume is just immense, and something that you're doing, you're crossing several fields here. How do you best keep up with
when it comes to the clinical stuff? It's very fortunate because there's a lot of you know, professional organizations who sort of collate the research and come up with guidelines and recommendations for the broad group of surgeons, right? Because not everyone is going to be someone who can assess the literature as well as, you know, someone who's trained in science. And certainly I wouldn't say that I'm, after this, you know, degree, qualified to judge all research and assess all literature, right? I'm probably better than most of the physicians who don't have advanced degrees, but someone with a degree in, like, you know, clinical epidemiology would know how to assess some of this research better than I would. So that's part of it. For the clinical stuff, you rely on these professional organizations to a certain extent. But there are, you know, you pick a couple of journals that are very good and topical, and you read their review articles, you read their editorials, see what the editors have to say, and that's how you sort of keep abreast of the literature. I have a couple of things set up where I've picked out some journals, and you can subscribe to their table of contents. So they'll every month or whatever email you a table of contents, and it has all of the articles that they published and like, you know, a tagline sort of thing. And you read through that and you see if there's something that is relevant to you, and then you can dig deeper. But it is a challenge, and like honestly, everyone has to try to figure out their own way of keeping on top that works for them.
Yeah, well, at least you have access to it. That's the important thing.
Mm-hmm.
This was one of my pet peeves is people who are not either highly degreed, or affiliated with universities don't have access to much of the literature for, I don't know, 3 to 5 years until it sort of kind of bleeds out there a little bit.
Yeah, I mean, Sci-Hub is such a great resource. If the viewers aren't familiar, but it's, so you should Google it because it's like domain changes frequently, but SCI-HUB, if you Google it, you'll come up with whatever its current hosting country is. And you can put in like the DOI, or like type in the title of the article that you found through like your Google Scholar or your PubMed search or whatever, and it'll pull it up for free.
What's the DOI?
So DOI is like I don't know, it's like a unique identification number for any paper that's published. So if you go on Google Scholar, for instance, and you type in your search terms for whatever you want to look at, usually you'll find an article. You may not have access to it, right? But if you copy paste the DOI from that and then put it into Sci-Hub, you'll be able to access. They have most articles for free, and you'll get the PDF. Yep.
Interesting.
Now it's very illegal for you know for like the people hosting it, but it's a great resource that, like, frankly, a lot of academics use. Like, I use it even though I have access to a lot of articles through my institution.
I've used academia.edu. Works to a degree. I mean, you've got
but that relies on the researchers posting their papers to
exactly. But that's one way to get around the publisher's limitations, because often I'll see the unofficial final version, which is like you say, them putting out their copy of it before it's published. Presumably there's no changes after that, and it generally seems to be that way. And if the articles aren't up, often you can just ask them. It's just there's an email link, and you can ask them, and you'll probably get it about half the time or more a week later or something. So that's been a good resource too.
Definitely. And I mean, you know, just while we're on the subject, the fact that, you know, some of these companies, like, I'm sure you've heard of Nature, the publishing group. They, you know, some of the highest impact journals in the world are Nature or Nature related, and to their charges for open access. So if a researcher wants to make their paper available to the public for free without a subscription, it costs the researcher $11,000.
Oh my gosh!
Yeah, it's absolutely crazy how much money it costs for, you know, and really the researchers are producing content for the publication. So it's a huge racket that
I don't
the
want to
publishers
get
are running.
too much of a tangent here on this, but is that a general problem of too much profit in that industry because of?
I mean, there's certainly an escalation, because now all the standards for researchers have risen to a huge degree, right? So everyone needs to publish, and you need to publish more and more to keep up with your peers. And so people are forming, you know, they're called predatory journals, and these journals will say, hey, they're sort of like pay to play. So they'll say like, hey, we'll publish your thing, just pay us, like, you know, $3,000, $5,000, whatever, and we'll publish it. It's like in our journal or whatever. So, you know, there's been a lot of research done on which journals are predatory, because you don't want your you don't want your article that you worked really hard on to end up in one of these predatory journals where it doesn't have the same,
Cachet.
rigorous review and
Yeah.
the sort of, scientific
That's
clout. So there's people who've done research on them, how to identify them. But ultimately, when you're getting into the more subspecialized your field is, the harder it is to know if you're getting into like a predatory journal or not. The big ones that everyone's heard of are also very general. They probably won't publish most research unless it's very, very significant for the field, right?
Yeah.
So it's a challenge.
This is a broader problem.
Yes.
Maybe we should talk about this on another show. Cause,
yeah, for sure.
Yeah, I mean, it's like, you see I don't want to say a layperson's article, but the clickbait kind of articles, like cancer, this discovered, is cancer about to get cured, kind of a thing.
Right.
And it's referring to some article. It's making it sound more exciting than it really is, and then there's no link to the original study, so you can't even get to an abstract half the time. So, okay, let's get into what the audience presumably wants to hear about, and that is cancer nanotherapy. So let's start going by what is generally, and you correct me as I'm going on here, because I'm not going to get into much detail, what is generally the route for cancer treatment these days. Because I've been out of this for a while. So, but the traditional thing that everybody knows about is chemotherapy, it's a drug therapy, radiation, and surgery, right? That's your traditional three-part method. Antibody therapy is something that has been probably 15 years back now has started. We've had that for a while. There's immune therapy. Immunotherapy, which is in part can be used to boost your immune system, your red cells, your white cells, and that in turn allows you to have higher doses of chemotherapy, for example. There is gene therapy to alter cancer cells, potentially. And I'll just kind of leave that there without getting into
I don't know.
detail.
I don't know of any gene therapies that are in clinical use personally, although I'm not I'm not a medical oncologist. Immunotherapies are a big thing right now, big deal. And they usually target very specific types of cancer. So the three mainstays are your surgery, chemo, and your radiation. You can make a distinction between solid tumors and sort of hematogenous tumors. So like your leukemias and your lymphomas are non-solid or like hematogenous tumors, which means blood, right? Blood-related tumors.
Explain lymphomas for
sure.
audience.
So leukemia, it means that it's a cancer of the blood, and lymphoma is similar. It's cancer, but it's referring to the lymph nodes. So we say that those typically originate in lymph nodes or in sort of lymph-related tissues or cell lines. But the thing that I think most people think about when they hear cancer are like a solid tumor, right? Like a mass somewhere. And for the most part, we say the only way to cure those tumors is with surgery as a primary modality. And that's becoming not so true as other types of therapy advance. But that's like sort of the broad rule. And then your chemotherapy and your radiation therapy act as adjuncts. So they add on and they sort of like boost the effectiveness, with the idea being that they will mop up any cancer cells that are remaining that have, if you've got a tumor in, say, your lung, you're going to remove most of the cancer cells in your body by removing that tumor through surgery. And then you're going to give like cancer and maybe a radiation therapy to kill the cells that maybe have disseminated around through your bloodstream, landed in other places. We know that tumor cells circulate. When a tumor is present, they circulate through the bloodstream.
Yeah.
Even if they're not metastatic and you don't have like another tumor somewhere else, they'll still be circulating through your body. So the chemo will mop up sort of that stuff that's still around, and the radiation does the same. So those two are considered the adjuncts. Then you've got your immunotherapy.
Sorry, adjuncts.
Yeah, go ahead.
Define adjuncts.
Oh, like something that you add on, right? So like if your surgery is your foundation, your main treatment, then you add on your chemotherapy and your radiation therapy. So like in a lot of cases, and again, this is changing, but in a lot of cases, you will do a surgery. The pathologist will look at the specimen that they've removed that has the tumor in it, and they'll say, yeah, they'll look at it in the microscope and they can tell from looking at what the cells look like if the cells look really aggressive, or if there's some invasion through different layers of tissue. And they'll say, oh, those are markers of more aggressive cancer. And so we're going to give you chemotherapy to help make sure that we sort of get all of the tumor. Whereas if it's a less aggressive tumor, a lower grade, we say, or if it hasn't invaded very deeply, then your medical oncologist might say, I think the surgery was sufficient. We don't need to do chemotherapy because that's just putting you at extra risk of having a complication because of the chemotherapy, for instance. So that's those three main modalities. And then you've got your immunotherapy, which is like a relatively new thing. It's very useful for very specific types of cancer. Like I know there's some big data coming out about like the effectiveness of certain immunotherapies for melanoma, which is a very difficult cancer to treat once it's metastasized.
Are immunotherapies the ones that are specific to an individual?
So they're not usually specific to an individual person. Some cancer treatments are very, very personalized or like precision. So they'll like, take your own antibodies or whatever out, and then they'll do something to them and then give them, reinfuse them. But I think, again, that's relatively infrequent. Most immunotherapies are things that you would, it would be a sort of a standard pharmaceutical, and then it would be targeted to a specific subtype of cancer that you might have, specific gene mutation, specific, cell surface receptor on the tumor, something like that. There's also like stuff like hormonal therapies for the hormone related tumors.
Oh, yeah.
So that's another
that makes
one.
Sense.
So like in breast cancer, there's Herceptin is like sort of similar to that, or tamoxifen, right?
Okay. So now let's jump into what used to be science fiction with nanotechnology.
Sure.
So the first thing that comes to mind, and I'll tuck away my pharmacist brain for a second.
Okay.
And we've seen movies about nanotechnology, nanobots. I think of literally the microscopic sized machines, tiny machines that can, and presumably they could be programmed to do something. And we see this in Iron Man's new suit, creating the new suit and all this stuff. Now, that's a completely different thing. So explain what nano-medicine is compared to that kind of sci-fi concept.
Sure. So why don't we break it down? So we've got nano and we've got medicine. And so there's a whole field of nanotechnology that exists external to nanomedicine, right? And for something to be considered nano, for nanotechnology, it measures between 1 and 100 nanometers in at least one of its dimensions. So you can think of a lot of things that have those measurements, right? And they would, qualify as nanotechnologies. And for some people are working on stuff like those nanorobots, nanites sort of things that can, do work, and they're on very small scales, and they probably don't have programmable chips or transistors or whatever in them, but they can manipulate them other ways. Like, people are creating nanotechnology that they can manipulate through magnetic fields, or, electrical currents or things like this. So that's one area. But if we're talking about nanomedicine, it just means that we're using any nanotechnology, so anything that's between 1 and 100 nanometers in one of its dimensions, and we're applying it to medical applications, right? So the treatment and diagnosis of diseases. So that's what we typically mean by nanomedicine, which is still very broad. There are a lot of things that qualify as nanomedicines that can be sort of like in vitro, so not something that you would put into your body. And so,
okay, outside the body. Okay.
Yeah. So there's a lot of like tests. So some pregnancy tests rely, for instance, on nanotechnology. They rely on like gold nanoparticles as part of their, how they work. And then there's, the stuff that you would do in vivo, or like you'd put in a person as a drug or a diagnostic agent. And so that's a different type of nanomedicine as well. And that's getting more towards the stuff that I work on. And I specifically work on cancer nanomedicine, which is using nanomedicines for, applications in the treatment and diagnosis of cancer.
Now, for scale, real quick, nano is about 1,000th of a width of a human hair, roughly.
That sounds right. So the way I think of it is it's bigger than most of the drugs that you think about, right? So most of the drugs that you think about are what we call small molecule drugs, right? Yeah. You would know this.
A couple hundred molecules, or a couple hundred atoms or something.
Right.
Yeah.
And so they'd be bigger in size than that, but they would be smaller than most cells that you would encounter. And it's actually the key feature of a cancer nanomedicine, or nanomedicines in general, is that they're at a sort of like biological, like a perfect biological size. And so what I mean by that is they're around the size to be able to interact with the cellular level mechanisms of the human body and of biology in general. So if you're too small, if you're on the atomic scale, cells don't really pay much attention. You can't really affect anything, right? And if you're too big, then you're getting into, well, you're too big. Maybe you're affecting tissue level stuff or organ level stuff, but you can't affect things on the cellular level.
So if it's too small, we think of things like how nerve cells work, where sodium and calcium and potassium go in and out of the cell.
Right.
And if it's too big, I'm thinking of vancomycin, or one of these chemo drugs that have just become chemical irritants to the tissues.
Sure. Or like, even surgical interventions can be, on that level. We're dealing on the, tissue and organ level. Surgery happens on sort of the millimeter to centimeter scale. And then below that, you're getting into the nano territory, where you can affect things on the nano and the micro scale.
Okay. Now for immunotherapy, that's something like immunoglobulin. These are very large molecules. These are 20. Yeah, my mind remembers 20 to 50,000 atom kind of size. So nanoparticles are going to be larger than that, right?
Yeah,
as we're going to see, I think.
Yeah.
Now, are most of the nanoparticles on quickly researching, but not researching, making one Google search.
Yeah.
Are carriers for drugs, or is it broader than that?
So this is really interesting. One of the things that is a bit unique to nanomedicines is they've invented a class of pharmaceutical, I don't know what you'd call it, called theranostic. So from that, you can see it's got therapeutic and diagnostic in it. It's a portmanteau of the two words. So a lot of nanoparticles and a lot of nanomedicine has focused on developing these theranostic capabilities. So a 2-in-1 solution to some of these problems, where they can do diagnostic applications as well as therapeutic applications. And both of those can be variable. So for instance, some of the original nanoparticles that we talk about are gold nanoparticles. And they are interesting because the size, so the number of gold atoms that you put together and the size that it produces affect the color of the fluorescence that it produces. So if you have a gold nanoparticle of a certain size and you irradiate that nanoparticle with like a laser, it will fluoresce in a certain color. And then if you add more gold nanoparticles to it, it'll fundamentally change the physics of how that fluorescence is created, and so it'll fluoresce a different color. And so you can use that.
What does that tell? What does that tell you besides the location? Does it tell you anything about the severity of the cancer?
No, not necessarily. And people use it for things other than cancer, right? So it's really fundamentally, it's a question of, what potential problems could we apply this sort of solution to? So I'll give you another example. There's a lot of nanoparticles that include fluorescent properties that don't necessarily change the wavelength or the color that they fluoresce, right? But if you can deliver those preferentially to tumors, for instance, then you can shine a laser on a field, right? On, like, say, a whole lung, and then only the areas of tumor will fluoresce, theoretically, and it'll highlight the areas of cancer, and that's a diagnostic application of a nanoparticle. And so this is one of the other sort of key features of cancer nanomedicine that really sort of blew my mind when I first heard about it, was nanoparticles, as a direct result of their size, will preferentially accumulate in cancer or in tumors as opposed to normal tissue. And there's a whole bunch of different mechanisms behind why it does that. But the bottom line is you would get, an accumulation of nanoparticles in tumor tissue, which means that's sort of like or at least we thought it was a real magic bullet, right? That's always been the challenge with cancer therapy, is we need to kill only the cancer cells and not the normal tissue. And so people have tried for years and years to find a way to target cancer cells directly. And it's not easy because, to your immune system, cancer cells often look just like your normal cells. They arise from your host cells. So in a lot of ways, they're very difficult to distinguish biologically.
So that enlarges because the chemotherapies, which are not specific to a particular to a cancer versus non-cancerous cell, they generally work on cells that are dividing. Correct.
Right.
So
so that's one of the mechanisms that chemotherapies use to try to be specific, right? Is they try to target, processes or things that cancer is doing that normal cells aren't doing as much.
All right. So the cells that are dividing normally in your body are your hair follicles, thus the hair falling out during chemo, and the gut lining,
right?
Thus all the nausea and stuff that comes with it.
Yep.
So helping to give the effect at the cancer cells allows people to have higher doses, right?
Right.
And reduce side effects.
Right.
I might jump to a graphic real quick so we can look at these beasties to see what we're talking about. So now in we don't have to get real specific in all these, but these one here from, especially on the top, you can see these all seem to be carriers. So tell us what you can about a couple of these that stand out to you.
Sure. So I guess figure A, the first one, this looks to be some sort of nanoparticle that contains drugs. So this is often, this is something that a lot of nanoparticles do. As we said, nanoparticles will preferentially accumulate in tumors, which means that you can deliver higher doses of your chemotherapy, for instance, directly to the tumor rather than letting it circulate through the body and cause your off-target effects, your side effects. And so a lot of nanoparticles will enclose your sort of standard chemotherapy drugs within a larger spherical structure called a liposome, for example. And the liposome will act as a delivery platform or a carrier that will preferentially deliver those drugs to the tumor. And they have
liposome is a fat, right? So
right. So
it's
if you
a
look
polar molecule.
in Figure B, that's a liposome. So there are two. It's a phospholipid bilayer, as you can see, and a phospholipid is has a phosphate head and then two lipid tails, which is a polar molecule. So the phosphate head portion is hydrophilic or like water loving, and so it will if you put a phospholipids into an aqueous solution or a solution that's sort of watery, they will form the lipid tails will align with each other, so they're not exposed. They're protected from any exposure to water, and the phosphate heads will line up so that they are exposed to water because that's where they want to be, and they'll produce these sort of hollow balls in a lot of cases, which is a phospholipid bilayer, and that's the same thing that your cell membranes in most of your cells. That's very similar to how they look, right?
This is what reminds me of origin of life research, right. And some of the earliest ideas of how cells may have initially come together, their cell wall structures at least.
Yep, very, very true. And so scientists will load drugs into the center of them, usually chemotherapy agents. If you look again on A, there's a thing that says PEG beside it. That is a long molecule that helps protect the nanoparticle from being digested by the host immune system. So it prolongs the circulation and acts to sort of shield the nanoparticle from the immune system. And then you've got targeting ligands. So if there's a tumor that overexpresses a particular protein or surface receptor, that will help target the nanoparticles to be even more specific to the tumor. And so the goal for a lot of these is just to really concentrate all of the drug in the tumor while limiting all of the any accumulation of the drug in any non-cancerous tissue.
Yeah, I like that concept in A of the ligands, the targeting ligands, because we've learned this from COVID, that the COVID virus has surface proteins, right, that are of various types, and they mutate. They can mutate to become different types of proteins, but giving us the ability to, once we have the ability to design the protein grabbers, essentially, right? It's kind of this lock and key thing. It's designed to fit a certain protein that would be found on. Is it more dominantly on a cancer cell, or do they have to?
Yeah, I mean, it's usually upregulated. So this is like part of the tricky thing in the balance is, so for instance, a lot of tumors will overexpress folate receptor, which they use to take in folate because, as you said, tumors do a lot of growing and they, require a lot more resources to continue their high rate of growth. So they upregulate all of these things to help them pull materials in, raw materials, so they can divide faster.
That's folic acid.
Right. Folic acid, folate, tomato sort of thing.
Yeah.
And so a lot of tumors will overexpress a folate receptor, which is like the catcher's mitt, right? And they'll try to grab as much folate. And so a lot of nanoparticles will try to take advantage of that by adding folate particles or folic acid particles, molecules, to the exterior of the nanoparticle to help it catch on to the tumor cells and get internalized. Of course, at the same time, that's only going to be useful for those cancers that overexpress folic acid receptor or folate receptor. So you have to have a balance between, well, am I going to make this so specific that it only works for one thing, versus, having something that doesn't work at all because it, is not specific enough.
Yeah, that's kind of a question for the pharmaceutical company in terms of trying to determine the size of their market, their target market.
Yeah.
how are these? If we have these molecules that are essentially they're not molecules, they're structures at this point,
Mm-hmm.
right? Once they become spheres, then you've
Mm-hmm.
got um, the drug inside it, or we had that one which was, I think it was F there, which it sort of captures the drug at various points within it.
Mm-hmm.
Um, how does the body get rid of these once you put them in the body, and presumably they hit their target and expose the cells to what the drug is? How does the body then remove these?
Yeah, I mean, the part of the issue is that nanoparticles are so varied, as you can see. So some of them are what you'd call organic. So they're composed of, typically carbon, nitrogen, oxygen, phosphate, hydrogen, these sort of things. And some of them are inorganic, which mostly means that they're made of metals, like heavy metals, like gold is a very common nanoparticle, silver are common nanoparticles, palladium, platinum, these things. The organic nanoparticles are typically broken down. So if you take a look, a lot of these nanoparticles will have a sort of molecular level structure for their monomers, and then that will assemble into a larger structure, or like a superstructure, right? So if you go to, say, for instance,
What's a monomer?
so if you pull up that diagram of the liposome, so if you look at B, you can see that so this is a liposome, and it has this phospholipid bilayer, which is again the phosphate head and the lipid tails. And so each of those phospholipids, the phosphate head and the lipid tails, is a monomer. And when
Oh,
you put a
like
whole bunch of monomers together, they will form this superstructure, which is the
okay.
liposome, right? So there's sort of like the whole is greater than the sum of its parts situation.
So I can see that a little more clearly on E, where you got the individuals,
Right.
right, that
Yes.
align with the hydrophilic on
Right.
the inside and hydrophobic on the outside. I think
Right.
that's right.
So that's a little bit different, right? Because that's not a bilayer. So, but
Oh,
it's
yeah,
illustrates the same point, right? So you've
simpler.
still got monomers there that are constituting, they're coming together to form a greater structure. So typically, these nanoparticles will be broken down. The monomers will be pulled apart through your standard sort of cellular processes. They get taken up into cells, into endosomes and lysosomes, broken up the same way that anything else would be. And then they get, metabolized and either excreted, if they're very small, through into the urine, through the kidneys, or if they're larger, through the gastrointestinal tract, in your like bile, essentially through your liver.
So broken
Um,
up by largely enzymes in the blood or by activity in the liver.
so usually not enzymes in the blood per se, but like enzymes in your macrophages. So like your immune
Ah,
cells, a lot of them are taken up by. In fact, that's like where most of the nanoparticles you inject go, is they'll go into the macrophages that live in your liver or in your spleen, and they'll be digested and broken down there. Um, when you talk about inorganic nanoparticles, so the metals, those tend to stick around, which is part of the problem with the
mm.
inorganic nanoparticles. They'll accumulate; they don't really go anywhere. So, like, the human body is really good at processing organic molecules. We can excrete those no problem, but metals we're not so good at excreting. So this is the problem with diseases like hemochromatosis, which is accumulation of iron, and it will
Mm.
sort of sit in your liver, sit in your heart, or Wilson's disease, where you accumulate copper because you're lacking
Copper,
some
yeah.
enzymes, right? And so that will again build up in tissues and cause problems. So the inorganic nanoparticles have a more difficult time being excreted and metabolized, and as a result, they're considered a little, sort of less safe than the organic nanoparticles are. So it's really hard to draw distinctions, but ultimately they're processed in much the same way as any drug is.
I'm wondering, this just off the top of my head, so I may be just totally wrong, but are are any of these used or clear out? Are any of these capable of like collecting up lead or something like that and pulling it to the
Like as
liver,
chelators, I
something like
don't,
that?
not that I know of.
Okay.
But some nanoparticles are used specifically for their ability to accumulate. So, for instance, there's one called ferraheme, I think it is, and it's an iron oxide nanoparticle, but it was originally used for treating anemia. And so they originally approved it just because people needed iron supplementation. So they approved this. And then they also found out that, hey, we can use this as an MRI contrast agent because it is, again, nano-sized in its formulation in the way it's constructed. And so it will preferentially go to tumors, so it can help us highlight areas of, tumor or areas of cancer, and we can detect it with an MRI. So there are some existing drugs that take advantage of that, but I haven't heard of any that chelate. It's theoretically possible, though. There are, there's essentially no reason you can't make almost anything into a nano formulation to try to give it a little bit more targeting.
Okay, I'm gonna bring up. Well, I've got one last slide that I love it because it's colorful. It's got a lot of cool shapes on it.
Sure.
Rather simplistically, but it shows the variety, just the sheer variety of types here, and I'm kind of fascinated by. Was it the carbon one on the bottom? It's like, is that a tubule or?
Yeah, carbon nanotubules. So I don't know what people use carbon nanotubes for. So they're not used in clinical practice for anything, as far as I know. The thing that we haven't talked about that some of these are used for is you can do a lot more than just deliver drugs with them. So we talked briefly, you can use them as fluorescence agents. You can use them as contrast agents for basically any imaging modality that you can think of. CT scans, X-rays, CT is the same as CAT scan, MRIs, PET scans, all of these things, like even ultrasound scans, you can use them as contrast agents for. And then you can also use them for different types of therapy. So there's a lot of therapies that we do where you can shine a laser and it can produce heat or oxidants that help you kill tumors, or you can do the same sort of thing but with an ultrasound, and so you hit them with ultrasound waves and it produces a lot of heat that helps you kill the tumors.
So that allows you to get the heat exactly on spot.
Right, exactly on spot, and very, very, very localized so that it only burns tumor cells. But yeah, this is a very good illustration of the fact that, basically anything can be a nanoparticle as long as it fits in that size category. It's a very heterogeneous group of sort of particles and technologies. But that's interesting. I haven't studied carbon nanotubes. I'd be interested to see what they're used for.
I'm curious, because this is kind of a fascinating thing too. Do you have any knowledge of quantum dots there? That one just seems to stand out as a whole different thing.
Yeah, so quantum dots are they're the ones that are typically they're the ones where you vary the diameter or the size of the nanoparticle to affect its fluorescence properties. So those are typically very small. They're sort of on the order of like single digit nanometers, so like 1 to 10 sort of nanometers in size, and they will they take real advantage of the difference in the way the electrons react when you're at such a small size, and a lot of that is fluorescence related. So they're taking advantage of the fact that smaller nanoparticles, smaller quantum dots will produce a different wavelength of fluorescence than larger quantum dots will. And people again use that so that's like what some and sorry, not just fluorescence, but like even visible light. So that's what a lot of some pregnancy tests are based on as well, is quantum dot technology. A lot of in vitro applications are useful for quantum dot technology. So things where they, take a sample of your blood or some other fluid or tissue, and they run it through a mechanism of some kind, and it reacts with the quantum dots in a particular way to change their size. And as a result, it changes the color that they fluoresce or the color of visible light that they reflect. and that in turn, helps you make a diagnosis.
Definitely more diagnostic than therapeutic then.
Yeah, typically.
I want to jump to questions soon, but let's see if I have anything else I want to ask you. Give me a general statement about state of where we're at. How effective are these, and how much of a jump forward is this in cancer treatment these days?
So the nano-medicine field has been struggling for 10, 15 years probably to find a foothold, because so there are some cancer nanomedicines on the market, like Doxil is one that's used. It's a liposomal form of doxorubicin, which is a chemotherapy drug. Most of the drugs that are on the market are sort of liposomal formulations, so they're just taking chemotherapy drugs that exist already and they're packaging them up so that they have some, preferential delivery. And so they help mostly with side effect profile. So they reduce the side effects a lot, which means you can deliver more drug to the patient and theoretically improve the overall effect. Although a lot of the studies haven't proven that you can actually significantly increase, for instance, survival over 5 years, but you can significantly reduce the side effects of the drugs. I think there's a lot of potential in nanoparticles, and the problem, I think, is that this preferential delivery is done through what's called the enhanced permeability and retention effect, or EPR effect. And basically, scientists have thought that this was very, very effective. And when we do it in mice, for example, we put a tumor in a mouse, the difference is shocking. They accumulate the nanoparticle, 10 times, 100 times more than any other tissue. But it's a much more subtle difference in humans for whatever reason, possibly because our cancers grow a lot more slowly because our overall metabolism is slower, right?
Yeah.
And so there's been a lot of problems with translation, right? Translating from the bench, from the lab to the bedside, to the hospital, to the actual patient.
From the animal studies
right.
too, like you just said, to the human. Yeah.
Right. So there's a lot of potential, but we have to figure out how to make that jump. And unfortunately, making that jump is it's a very expensive and risky process, right? So I'm sure you're aware the typical number quoted to bring a drug from like its discovery in the lab to an actual marketable drug that you can buy off the shelf costs $1 billion.
Oh wow, I didn't realize it was anywhere near that high these days.
It's really high. And it's even more difficult for nanoparticles because of the regulatory mechanisms that are in place. So the FDA was founded predominantly to regulate small molecule drugs, and so a lot of their testing and their quality control measures are tailored towards that. And their tests that nanomedicines have a hard time passing just because of the nature of how nanomedicines are created. And so there's a lot of problems with the regulatory bodies, with the deck sort of being stacked against nanomedicines. There's a lot of problems with the mechanisms that nanomedicines use not translating super well. And so it's still a lot of development and a lot of discovery going on in what the actual best way to use nanoparticle technology is in humans. And so this is like, I think people have been trying to do this this same thing where we are trying to deliver drugs using a nanoparticle carrier. And I think that's actually the less interesting potential application.
That's what I'm struck by, yeah.
So one of the things that I did a lot in my studies was, so I did some fluorescence work, and I think that's gaining a lot of interest as the technology improves, the like imaging platform technology. So now we have cameras, like laparoscopic cameras, like long sort of telescoping cameras that have a fluorescent laser and a fluorescent, lens and like detector, and we can now use those to detect nanoparticles. And so you don't need to have a 10 times, 100 times difference in the concentration. You only need to have like a 2 times difference in the concentration,
wow.
for instance, to be able to detect it and to find that threshold. Similarly, one of the other things that I did is something called photodynamic therapy, which is where, and I alluded to this, you shine a laser at the tissue, and the nanoparticles that have accumulated there will produce reactive oxygen species or oxidants. Some people might know them as oxidants. So, like you eat blueberries because they have antioxidants. Well, we want to deliver oxidants to tumors because they help to kill the cancer cells.
Because too much oxygen will break cells down, break molecules
exactly.
down. Yeah.
And so with something like that, you've got 2 layers of selection, right? You've got the nanoparticle, which to a certain degree accumulates preferentially in the tumor, but then you've also got the laser, and you're only going to cause an effect wherever you shine the laser. So if you know what the tumor is, you can just shine a laser on it and it'll sort of kill the tumor preferentially, and you don't have to rely on that mechanism that doesn't work so well in humans as we thought it did.
This is going to be an interesting field for a long time, because it seems like there'd be so many, at least especially hypothetical ways to treat, and like you say, it's going to be a matter of largely getting the concept to the actual reality in a living being, right? It's like it's like talking about how easy it is. It would be easy to go to Mars, but it's difficult.
The feasibility aspect of it
Yeah.
is different from the technical possibility of it.
Right. Yeah. When you get down to how to keep a person alive on the way there.
Yeah, absolutely.
Okay. We got a couple of questions in the chat, so I'm going to pull these up. If anybody else has one, now's the time to add to that. I've got one from Aaron Salas. Can MD speak about whether his field of study has suffered similar issues with replication crisis?
Yeah. So I think the replication crisis that he's referring to is, a lot of studies have shown a phenomenon, and then when someone goes to try to replicate that by following the same methodology used in the original study, they can't find the same thing. And so you think, well, what does this mean? Was it just like a statistical quirk that happened in the first place? Was there something else, another factor that we didn't consider, didn't write down? I think that the replication crisis that nanomedicine has faced is, again, this issue of not being able to replicate the effect that we see in animals, particularly in rodents, which are cheap, high throughput sort of models, right? You can, like, I've used hundreds of like rodents in my studies, and it works great. Nanomedicines work great in rodents, but the replication in humans is more difficult. The replication, even like larger animals, is much more difficult to do. And so I think that's the real replication crisis that nanomedicine faces. I think the replication crisis in a lot of other fields, I think it's most predominant in, like, say, the social sciences and psychology, for example, where there may be a lot of, factors related to the current culture and the current circumstances, and like maybe the sampling of the population and things like this that were not appreciated at the time. But in retrospect, you can think, well, those are probably the reason why the results came out the way they did. But it's a very good question.
There's no mandate to replicate.
No, so that's a problem. And in fact, there's very little motivation, because no journal is going to publish. That's the currency of academics, of researchers, is publishing papers. And so no journals are only interested in something if you're doing it for the first time. It's all
Yeah.
about novelty. Can you, can you show a new phenomenon? Can you apply a method to a different, problem that no one's done before? That sort of thing. But there is no motivation. There's no, remuneration or no value to replication built into the system, unfortunately.
This would be a nice. I personally think we do need a cultural change, particularly to replicate to publish negative results. For example.
Yeah, I mean, so there's a whole bunch. That's a huge, almost separate issue, right? Is
Yeah.
what you do with negative results, because it would be a good thing to have a sort of journal of negative results,
Yeah.
where everyone could just deposit the things they've tried, right? Because science right now is a selection bias where only the things that work get
Yeah.
published, which means that maybe there's 10 researchers who try the same thing and waste a whole bunch of research money and resources and all this stuff, following the same, going down the same rabbit hole that
Yeah.
that's not going to work. No one's
Yeah.
ever published that it doesn't work, right? And why would they? It doesn't make for very compelling reading, but it's something that we should make some sort of international,
Yeah.
deposit or repository of things
Yeah.
that don't work. But again, who's going to fund that? Who's going to
Yeah. I was
run
gonna
it?
say the funding's got to be a part of that.
Right.
Okay. Aunt Jer has a question. This is going to be an easy one. Are mycelials what they use in microcellular water beauty products? And
Yeah.
what do they
So
do?
I've vaguely heard of. I mean, I don't use a lot of beauty products,
Neither do I.
so I don't know anything about micellar beauty products. But micelles, I imagine, are like sort of the same idea. And I think what they're trying to go for is, if I had to guess, is that the micelles have this sort of have this polarity to them, right? So they have a lipid component and they have an aqueous component, or like a hydrophilic component, which means that they'll be good at dissolving potentially both hydrophilic things that you can wash off with, say, water. But they'll also be good at dissolving and removing lipid-based things, so like grease or oils and things like that.
Yeah.
I'm inclined to think the skeptic side of me, my reflex is that these sort of beauty products are a bit scammy, but I'd have to do
Yeah.
a deeper dive into it. It's a very interesting question. Maybe I should look into that, actually.
I kind of expect that too. Although I will say, from my pharmacist side,
Mhm.
the basic difference between a cream and an ointment is oil and water versus water in oil.
Right.
So a cream, for example, is where you have oil trapped in a water bubble, right? That causes an emulsion, so it becomes less greasy. You can
Mhm.
still deliver the oil to the skin or whatever it is, but it's less greasy,
Mhm.
as opposed to the other, which is oil on the outside, water captured inside. You're
Mhm.
delivering hydration, but it's just like Vaseline,
Yeah. Yeah.
So okay, and one more. Does MD Aware know
Almost a
almost a
doctor from
doctor, Matt
Australia.
from Australia? He mentioned lasers at the end of laparoscopic.
Yeah, laparoscopic cameras.
Thank you.
I don't know almost a doctor from Australia. That sounds cool. Aunt Jer, you should email me his details, and maybe we can have him on Truth Wanted or something, or on Skeptic Haven.
That'd be cool.
Yeah.
Well, that cycles us back to the skeptic secular world. Where can we find you? Where can we find you out there? I know you've said Talk Heathen.
Yes. Truth Wanted. Truth Wanted is my home show. So I'll be on actually this Friday. So tune in and call in if you want to talk more nanomedicine, laser stuff, or anything else. You can email me, md aware at atheist-community.org, or you can tweet at me at EMDEE aware. So MD aware, but spelled out. So you can find me those two places. Like I said, I'm on Truth Wanted this Friday. I might be on one of their other shows towards the end of the month, and I'm on every so often. So I'm around.
And to protect you a little bit here, how much do you want? Are you willing or do not want people to contact you with medical questions?
So I'm not going to provide any specific medical advice. Go see someone who can see you in person and examine you and knows your medical history and all that stuff for that. But I'm happy to talk medicine and science in general terms with anyone.
Well, we all push science, and I'm very curious to catch you at some point on an evolutionary biology discussion.
I'd love to do it.
So maybe we can, the next time we have an evolution panel, Wes, take note and invite you into that. That would cool. Be kind of cool. Do you find the last question that comes to mind is: do you find doing the skeptic and secular stuff as sort of another interest, or do you find it sort of a relaxing change of mental part of your brain, or
I like having different? These sort of discussions, like, you know, the sort of like debate discussions. It brings up it. I think it goes along with the fact that you want to challenge your beliefs, right? And so most people, you know, you start by, if you want to challenge your beliefs, you start by doing it to yourself, right? And you sort of challenge your own beliefs as much as you can, but you're always going to have blind spots to it. And that's where having these sort of like debates, confrontations, discussions comes in handy, because hopefully you end up talking to someone who has a wildly different opinion and wildly different reasoning than yours, and maybe they can reveal something that you can, you know, use to develop your own beliefs further and sort of lead you closer to the truth. And so, I mean, again, if we're talking science, this is the point of like scientific conferences, is to go and talk about stuff and challenge each other in you know, a more sort of intense forum. And this is the same thing for me. I just want to, as we say, I want to have as many true beliefs as possible and as few false beliefs as possible. And I feel like to a certain extent, I've gotten as far as I can go with just thinking about it myself. And so I want to talk to people who disagree with me and see what they think and why. And so I also find it fun. So
cool.
It works every way.
That's a piece of it. It has to be a piece of it.
Absolutely.
Well, thank you so much for agreeing to, you know, do something a little different and getting into the medicine this time.
Yeah, it was great fun. So yeah, this is like the thing that I'm actually qualified to talk about, right? So that's nice. All that other stuff is just, you know, I may have the MD or whatever, but it's just my opinion for the most part. But this is like a thing I'm actually really qualified to talk about. So it's fun. It's a nice change of pace.
Well, I'm glad you came.
Yeah, so, thanks for the invitation. It was great.
I want to thank our Wes, do you have a slide for me on upcoming shows that you want me to push, or while I'm saying some of this other stuff, you can pull that up if you have it. Want to thank the other producers of Skeptic Haven. Producer Wes, I think everybody in online knows producer Wes. Kat the Humanist, Jena, Miyu. I'm gonna miss some people, so I'm sorry about that. I don't have our list of patrons handy, so I'm gonna have to skip that right now. We do have a new patron that I want to give a shout out to, and that is Kathy Cotton. So thank you, Kathy. That's Aunt Kathy too, I believe. Like and subscribe again. Check out our Patreon. MD, you said you had your own channel?
I don't have my own channel. All my stuff is through ACA or whoever invites me on their show.
Okay.
So, but you can tweet at me. That's the most direct route to get to me. Good to know.
So I hope this was not too specific, because it's, you know, I was excited to learn about what this next sort of field of treatment is. So, and I think it sounds like from what you said, there's a lot more to come over the next couple of generations, probably.
Yeah, I think the next 10 to 15 years is going to be big.
Nice, cool. That's good for all of us. Well, thank you everybody for tuning in, and we will see you next week on The Sunny Spot.